Started. Can you see my screen?
Yes.
Okay. Welcome to all the students. Here, Professor Pramod Wangikar and myself, Amit Sethi, we will give brief information about the work that we do, and then we will take your questions about KCDH. We also have staff from KCDH present here, Mrs. Smita Wangikar and Dr. Rashmi Tiwari, and they have been the backbone of setting up and getting KCDH running in its first year. So I'll briefly talk about the work that I do, which has to do with digital pathology. So when we have uh, tissue slides that has to be seen under a microscope, if instead of looking at it in a microscope, it is scanned using a specialized scanner, you get a very large image, which is sometimes a gigapixel image. And now, if that image is available, we can use data science such as machine learning, deep learning, image processing on it. And based on that, we can automate some of the work, such as finding cancer in tissue, grading cancer, finding different subtypes of cancer, and so forth. So that is what my research group does. And we closely collaborate with medical doctors, such as those in Tata Memorial, University of Illinois, Chicago, and King's College, London. And a lot of students are involved, right from undergrad to dual degree to masters and PhD, who work on this type of data. And because it's real world data, it's not a standard data sets that we work on. We work on data from hospitals. The data can be messy, and that's where the challenges lie. The data can be scarce, and especially when you think about machine learning, you need labels to learn from. You may not have good labels all the time. So those are some of the problems we try to solve. So we do things like replicating what a human would do, what a pathologist would do, and then we also develop algorithms that go beyond human capability, and then try to overcome some of these challenges, and also estimate uncertainty in the AI or in the deep learning algorithms that we develop. I'll explain what this means. So this is a typical cycle of cancer treatment, and at every stage, imaging is involved. So if we have uh, uh, if someone is suspected of having breast cancer, then there would be a diagnosis. In during diagnostic process itself, we will have mammogram involved, MRI involved, biopsies, chest X-rays, etc. involved. So these are all images. And once the cancer is diagnosed, there would be some neoadjuvant therapy, which means we would try to shrink the cancer before surgically removing it. And in neoadjuvant therapy, once the chemotherapy drugs are given, then we try to assess whether there is any response of the drug. So that involves more imaging. And after that, we give adjuvant therapy, which means we first surgically remove the tumor and then give another round of chemotherapy or radiotherapy. And after that, again, imaging is involved to see whether um, the cancer has completely gone or not. So this is a some sample of a tissue slide at high magnification. So these black dots that you see are cell nuclei, but this is not the natural color. There is some dyes that are put on them. They are called stains. So you have purple and pink combination, which is quite common in pathology. It's very inexpensive to use this hemotoxylin and eosin stain. So the holy grail is that can we use this inexpensive stain dyes that are prepared using that, but then use deep learning uh, to classify between, let's say, cancer cases versus control cases that have no cancer and so forth. Um, there are more expensive imaging techniques available, uh, but they are not accessible to a large swath of our population due to its cost and it not being available in every hospital. So we are trying to develop these algorithms for inexpensive uh, images. So here you could classify between malignant, which is cancerous, versus benign, higher grade versus lower grade cancer, more dangerous cancer versus less dangerous cancer, cancers with certain mutations that have for which certain treatments are are to be used versus those that don't have the mutation, and those that will respond to a particular treatment versus those that will not respond, etc. And then also predict survival. So this is like a roughly increasing order of difficulty of problems. And when we develop these algorithms, they have to be tested on separated data so that we can see that they will work well in the real world. So at the very least, we should have, even if we have a large image of one patient's tissue, we should not take some portion of it for training and another portion of the same patient's tissue for testing, because then your algorithm learns patient specific characteristics as opposed to disease specific characteristics. So we should have at least patient level separation. Uh, Stronger level of generalization we can test if we have hospital level separation. That means you train on hospital A and B, but you test it on hospital C. And then population level separation and data acquisition, which means that you are using different brand equipment such as GE versus Philips, MRI machines, and see that it generalizes from even GE and Philips. So this is, no one is doing this, and I don't think we should jump into this right away. If we can do at least hospital level separation, that would be good enough for now. So this is an earliest example of the work that we did, which is we are trying to replicate a human expert. So this is a different dyeing scheme, a different type of a stain. It's called IHC stain, HER2 IHC stain. And here you can see all the nuclei are blue, and then you see that around the nucleus, sometimes you have a brown complete circle, sometimes you don't have it. So when you don't have it, then the tissue is not mutated with the HER2 mutation. If you see the brown circle, that means it is mutated because it is sending those protein out, and then to that protein the brown dye is binding. So we want to detect whether the cancer has HER2 mutation, and because there are specialized treatments for that available. Now you cannot simply look at the color, because the color intensity of the color can be misleading. You can see that this is the one of the 2+ patches is more intense brown than the 3+ patches, uh, and that can create a problem in if you just use color. So we have to use more in uh, non-linear algorithms such as deep learning and so forth. So we developed one, and it worked very well on one particular data. Another challenge is that when you get data from two different hospitals, sometimes the colors look very different. And if you train ML on data from this particular hospital, it may not work very well on data from the source hospital. So we developed a stain normalization technique where we don't change the structure of the tissue, but just change the color using sparse non-negative matrix factorization. Uh, then we developed a large data set of nuclear boundaries, because detecting and segmenting nuclei can be important for understanding what is going on in the tissue uh, by detecting different types of cells, cancer cells, lymphocytes, macrophages, etc. And based on that, we developed an algorithm, and we also open sourced the data set that a lot of researchers from around the world are using. Because these annotations, you see these green pen marks that you have, the circles that you have on the nuclei, uh, this kind of annotations are important for training these ML algorithms. Now, we use data sets from prepared by other researchers all the time for our own research. So this was our contribution of giving back to the research community, because such a data set didn't exist at the time, so that people can also, others can also benefit from it. Then later we also enhanced this data set with the different type of nuclear marking as well, and launched another competition. And certain labs from around the world, they did very well on the competition, and they developed algorithms that work very well. Um, so I was showing you earlier this IHC staining scheme, which is more expensive versus HND, which is less expensive. So now we are trying to detect mutations in HND itself because that is inexpensive. Uh, so we take tissue where part of the tissue has been diagnosed using the more expensive stain, and part of it is we have the image available for the less expensive stain. So there are subtle changes in the cell shapes and how they organize that actually can capture the telltale signs of HER2 mutation. But a human eye cannot reliably detect it from across tissues. Or if a very expert pathologist can detect it, they cannot teach it to their junior pathologists. So here, detecting that using ML would be very helpful in reducing the cost of this test. So that's exactly what we did. We looked at these tissues, we marked regions that looked HER2 positive versus negative based on their neck section, which was stained using the IHC, the brown stain. And then we developed a three-stage pipeline, which one stage detects nuclei, another stage detects cancer among nuclei, and third stage detects whether they are HER2 mutated or not. And it did very well. Similarly, we did for another cancer and its mutation using something called weakly supervised learning, where we did not even use the markings of the regions. We just knew from a DNA test whether the entire tissue was BRAF mutation positive or BRAF mutation negative. Um, and we use a more advanced form of architecture of a neural network to not only detect the mutation but also the regions of the mutation, which gives us these heat maps. And based on that, we can discover what the cells look like. And this confirms what some pathologists long ago suspected BRAF mutation looks like, but they couldn't teach it to others. So we could confirm that without telling the neural network that how to detect it, where to look for. Let's give some of this. And the last thing that I'll talk about is robust AI. Here we are developing two kinds of solutions. One is if we assume in ML, whenever we train an ML model, we assume that in the training data, all the labels are correctly given, which is not always true with medical data. Because medical data is difficult to assign labels. There's continuum of disease states. There are no distinct classes like cats or dogs. Between grade 3 cancer and grade 2 cancer, there is theoretically a grade 2.5, but no one calls it 2.5. So the distinction is subjective sometimes. So there are differences in diagnosis between Dr. A and Dr. B. So that is one reason. The other is obviously it is not as straightforward to detect like different cell shapes and cell counts and so forth. So assuming that some of the data is mislabeled, how do we detect it in the training data and discard it during training? So we train a neural network for a little while, and then there is something called a loss function of a neural network, which is the objective that it is trying to minimize. We model that loss function using a distribution, and the sub images that have high loss, we assume that they are mislabeled and we discard them from future rounds of training. And that improved our accuracy and also gave us more informative sub images. The second type of uncertainty that we are looking at is once you have a trained method, uh, if you uh, if you deploy it in the clinic and the clinician makes a mistake in using it in the sense that it, so let's say the model was trained for disease A and disease B, but the clinician accidentally gives it disease C. So instead of forcing a decision between A and B, the model should say that this is not a data that I have seen before. It should detect outliers and it should discard those outliers. Another reason you can have an outlier is, let's say the model was trained on MRI data from GE, but now accidentally the clinic is using Philips and they are using the model, then it should say that the image characteristics look different and my model the decision should not be trusted. So we are developing techniques for that. We try to bring make tight clusters of each class in addition to finding a boundary between them. So now if you have tight clusters of class A and class B, if you have test data 1, 2, and 3, you can see that those that are within the cluster or close to the cluster center, those are will be classified as A or B, but those are that are far away from any cluster, they will be classified as outliers. So these are some of the practical problems that we are trying to solve in medical imaging. Uh, I can now hand it over to Professor Pramod, and then we can also take questions about KCDH.
Yeah, thank you, Amit. So I'll follow the pattern that you have used. So I'll also make a very quick presentation, and then we can go to question answer. Is my screen visible?
Yes.
Okay, so my name is Pramod Wangikar. I am a professor in chemical engineering, and our work is primarily on metabolomics. And this cartoon shows metabolomics that can be done on human samples, animal samples, bacterial, plant, drug samples, and so on. And the left hand side is a chromatogram of a given molecule, and right hand side is a mass spectrum of the same molecule with the mass spectrum obtained in the experimental sample is above and in the library is below. So based on library matching, one identifies molecules that are present in a given sample. So for example, in the blood sample, you could have anywhere between 500 to 1000 metabolites, small molecules, that you will see that you will be able to identify and quantify using a single assay. So what are metabolites? So this is just a bird's eye view of metabolism, and this kind of metabolism will be sort of common to all living systems, humans, bacterials, plants, and so on. The dots are metabolites and the lines are the reactions. So we are interested in the metabolism in general, and we are interested in identifying the metabolites and their relative concentrations. So the instrument that we use, LCMS, liquid chromatography and mass spectrometry, basically separates based on chromatography. So the one axis is retention time on the chromatographic scale. The other axis is mass to charge ratio, M by Z, on the mass spectrometry scale, and the height is the intensity. So you may have hundreds and hundreds of metabolites in a complex sample like blood sample, and you will be able to separate them. So the challenge is in data analysis. So our focus is on data analysis. So this is what we do: mass spectrometry and data science for healthcare applications. On one hand, we are developing software for identification and quantification of metabolites, where we use deep learning based algorithms to address some of the challenges in mass spectrometry data analysis. The data is quite noisy, and I'll show you some examples. The data is complex. The data has 3-dimensional, 4-dimensional, and even 5-dimensional nature because of the different dimensions in which one can separate the molecules. And I showed you 2 dimensions, chromatography and M by Z. And there are additional dimensions that one can see. And then an analysis of such complex data using statistical tools. The application areas are biomarker discovery, let's say for cancer, new biomarkers for cancer or pre-diabetes and so on, or applications in process development where the process itself involves bacterial cells or animal cells, diagnostic tests for new diseases. The next generation applications include sports and athletic training, elite sports, those kinds of solutions. Development of specialized flavors, plant-based meat, and specialized beverages. These are some of the applications that we work on. overview of data analysis, we start with instrument generated data and analyze it through the entire pipeline, which involves identifying the regions of interest, aligning the peaks, filtering the data to remove noise, identify the metabolites or the compounds, statistical analysis, and then at the end, a human expert needs to look at the data. So in my lab, there are several students who actually do the experiment. So they are from the biology sciences side. So they are the end users. And our objective is to also be able to produce tools that can become web apps for others, for the community, scientific community to use them. So overall, I can show one example of a very small piece of data from a very complex data set. So a sample is injected on LCMS and it generates close to 100 MB of data per sample. So a small slice of the data is shown here, and I can see that the particular compound gets separated in one dimension, one of the dimensions that is used for separation. But in the other dimension, it doesn't get separated. And of course, it has the same molecular weight, so it can't be separated on the mass spectrometer either. So this kind of data is quite complex. You need to make sure that the separation is true separation and not just an artifact of the data or the data analysis tool. And we develop those kinds of tools using artificial intelligence based techniques. Another application is removal of noise. So these are the chromatograms that are shown, and some of them are noisy. An instrument will pick up all these chromatograms and will present them to the user. And the user then need to separate the true peaks from the noise, which is of no interest to the user. So this itself removes about 90% of the data from the data file, and then the user needs to worry, or the subsequent data analysis steps need to worry about only the 10% of the data that is true data or good quality data. Another application is demultiplexing. So the instrument uses a multiplexing algorithm to improve the duty cycle of the instrument. So this is a hardware type of hardware-based multiplexing. The multiplex data needs to be demultiplexed and denoised. So we have developed a tool to demultiplex and denoise. So this data, which is basically 4-bit multiplexed, has got has 8 peaks here. So they got demultiplexed and only 1 peak is visible. This data has 2 peaks in it, and because of multiplexing, you could see potentially 16 peaks here, the 4-bit multiplexing, and then you could, after demultiplexing and denoising, you get 2 peaks here. And this is an example where it's that actually the molecules are separated in the chromatography. So when you demultiplex each of these, you get just 1 peak at 2 different retention time values. Then the data needs to be grouped and need to be reported in a transparent manner to the user. So this is just the reporting of the data by grouping and aligning and so on. So I'll not get into the details. And then one has to perform statistical analysis by using all the standard statistical analysis tools. The data needs to be visualized based on how the user is used to see the data normally. So the cloud plot and the volcano plot, they are generated. Then one has to also see the quality of data, do some kind of exploratory analysis, some kind of clustering, PCA plot, generate the heat maps, and so on. So all that is done to basically to get closer to the objective of data analysis. And the useful data is then presented so that it becomes part of the reports for regulatory agencies or for a publication and so on. So these are the kind of data that gets identified and finally plotted for a given sample. So this is another example. The applications, as I said, diagnostics and well-being. So diagnostic, of course, to diagnose the disease, and well-being for for people to live healthier and longer, and to get early warning signs of a disease if there is any. Toxicity assays of drugs, bioprocess development, pharma and biotech R&D. And in our lab, so my lab is a combination of experimental science and data analytics science. So the students, the PhD students who are into the experimental part, they are constantly collecting data on bacteria, humans, plants, and so on. And and so that will provide a complete ecosystem where the experimental scientists and the data scientists will work together along with external application experts as well as our collaborators within IIT, who help out on the data science and the machine learning aspects. So I will stop here. And we can take questions. Is there a moderator who wants to ask questions or?
Prapti?
Yes, ma'am.
So you can ask us questions about our work. You can ask us about KCDH. You can ask about the programs that we have, the minor program and the IDDP program. And there are some IDDPs that have been floated. We can try to answer questions about those that other professors will be supervising to the best. We can understand about what that project is about.
Right. So firstly, we can begin with those IDDP projects only because we have been discussing on that. So could you just give an overview of the other projects as well?
Sure. Let me take a look. Okay. Can you see my screen?
Yes, it's visible.
Okay. So this first project by Professor Pushpak Bhattacharya is about assessing mental health of patients using multimodal data. So many times a single mode of data is not enough for making a decision. So for example, for a patient, you might want to look at how they speak and how they react to certain stimuli, etc., to assess their mental health. So here they will try to develop ML models that will look at natural language processing, which is text, speech data, which is audio signal data, and computer vision, which would look at images or video, which is how the perhaps where the gaze of the patient is and so forth. So I'm taking a guess here. I don't know the details, obviously, but they will be combining these using something called multitasking neural networks, which means that the neural network is combining multiple modes of data as well as it is trying to meet multiple objectives. Uh, I think this second project is quite similar, except that the medical diagnosis would be different here. They are looking at different personality types, again using same multimodal data and multitasking neural networks. Any questions on these two? So it looks like they already have uh, publications that are accepted on this line of work. This is a project by Professor Devashish Chatterjee and Professor Neeta Kanekar. So they are looking at pathological gait in humans. So when there are certain neurological defects or neurological diseases such as Parkinson's, then the way humans walk, that becomes it doesn't it deviates from the normal way of walking, and that can be uh, if that can be caught, then it can be used for uh, early diagnosis of these diseases. So there are many ways of capturing the gait data. One is that you put sensors on the body and ask the person to walk on a treadmill, and then you look at their changes over time. Uh, the other way would be that because putting sensors on the body is difficult, then you just captured a video of the person walking on a treadmill, and then you develop computer vision based models to analyze that gait. So one way is to actually find all the joint angles in that video and then model the gait. The other is that you don't go into this kind of precise modeling, but you actually have diagnosis available for everyone for a training data set, and then you simply uh, have a deep neural network which is not very insightful, but it can directly classify from the video itself. This particular project, phase 3 randomized study to develop novel AI-based prognostic tool for PET CTs and pathology images. This is a collaboration between Dr. Hasmukh Jain, who's an oncologist in Tata Memorial, and Professor Ganesh Ramakrishnan from computer science. So this is quite similar to the kind of work that I was talking about earlier. So if you have images such as PET scans and CT scans combined, and or if you have pathology images, then can we develop deep learning models, especially if you have these images of patients who were treated long back and then now you know the outcome of the treatment. So can we now build a predictive model for that, for whether the treatment will be successful or not. So then that will help us prognosticate what will happen after the treatment and then help choose the right treatment. So currently, how the treatment is chosen is based on first grading what is seen, what kind of cancer is seen, and then how big it is and how it has spread. But are there other signs that can tell us whether the treatment will be successful based on things that have not been noticed yet by the pool of experts who have developed these protocols? Then Professor Deepthi Gupta has floated this project, Applications of AI and ML in Neurosurgery. So neurosurgeons, they rely on their clinical, on their experience and clinical evidence to make a decision about prognosis. So it is on similar lines that can we go beyond this human capability and look at retrospective data. Retrospective means past data, and based on that, can we make predictions about uh, um, the recovery of the patient after the surgery for future diagnosis. So this will also help them localize the tumors or seizure zones in using perhaps fMRI data in of the brain. Professor Pramod, please feel free to jump in case I misspeak about some.
Yeah, so what I was thinking was there are 30 projects,
Yeah.
so we might not be able to go through all of them, but broadly speaking, if I can jump in here,
Yeah.
broadly speaking, the dual degree project is a 12 to 14 month long project, runs into your 9th semester and 10th semester. So, primarily in your 5th year, and it's a research project. In KCDH, you will find projects that are connected to health in some way or the other, and there is a digital or a informatics component attached to it. And within that, again, there are two types of PIs you will see. So some PIs are primarily in the domain of informatics or machine learning, data science, and some PIs are in the laboratory or biomedical side. So I fall in the second category. And and of course, we have interests in developing certain tools or certain devices or certain tests that take help of digital tools or informatics tools, and we collaborate with those who have expertise in that. So typically you will have research projects that are in these ranges. Also, when you do a research project with one of these professors, you will basically be working with that professor, with their PhD students, their research group. So what is expected is that you will get a completely immersive experience of a research group, for which you typically need to enroll in a PhD program or at least in a master's program anywhere. So you will get that experience right here as part of your dual degree project. So that's the overall objective of this. Plus, a lot of these projects have a collaborator, external collaborator, either a clinician at a hospital, or a company, a pharma company, a diagnostic company, and such. So there will always be this chance to interact with that collaborator who is connected to a real world problem, and they are trying to solve a real world problem, or they are treating patients and so on. So that immersive experience will go beyond IIT, and it will depend really on the student as to how much they want to gain out of this dual degree project. And it's an excellent opportunity to apart from getting that additional orientation on digital health, and improve your prospects of getting job opportunities in certain sector. Apart from that, these are the additional advantages that each project will bring to you. And of course, there is this is a project that will be driven by self-motivation. So motivated students will also be able to publish papers and go beyond what is minimal required to earn the degree. So all these are the advantages I feel. And if any student has any specific question about the program or about any particular project, that we can try to take. And also, when we try to describe projects of other professors, keep in mind that we are not experts in those areas. So those professors are the best people, and the easiest thing for you to get clarity on those topics will be to write to the respective PI directly. Ask for a time for 15-20 minutes of discussion, etc., or just ask for queries online. You will get answers.
All right, sir. Thank you very much for that. And we have been getting another question that, like healthcare informatics, do students require any background in biology or computer science for that? Because not all students might have a strong base in that. So, is there any prerequisite that is required in the field of healthcare informatics, or at least some background in biology or computer science is required or not? Will you just answer that?
So the programs are designed in such a way that students with any engineering or quantitative background can develop that expertise through the course system. So for both minor and IDDP, you have to complete additional courses offered by KCDH, and those courses will cover basic healthcare related information as well as basic data analysis expertise. So you don't have to worry about prerequisites if you are already in the IIT system. We will give you the prerequisites in your when you enroll in the IDDP program.
Yeah.
Professor Pramod, do you have anything to add in that?
So I think he has already put it. And so there is a set of coursework that needs to be done as part of the dual degree in your 4th and 5th year. Some of it is mandatory courses and some are optional courses, electives. So mandatory courses will introduce you to the health informatics and physiology and so on. And the electives you can take based on what is the interest. If you have interest in machine learning, you can take electives in machine learning. Yeah.
Okay, thank you, sir. So, expanding on the coursework only, so like what were the aspects that the faculty had in mind while designing the curriculum for healthcare informatics? Was there any specific vision that they had to, which they targeted while designing the curriculum?
You want to start,
So
Amit?
yeah, so uh, like we mentioned, the goal was to cover both healthcare as well as informatics uh, components. So we have a basic course on physiology, which is a core course, and a basic course on analytics, which is also a core course. And then between these two, which one do you want to specialize in more? And then within that, there are sub specializations. For example, there are courses on different aspects of healthcare as well as informatics. You can specialize, for example, in image processing versus some of the omics courses, the uh, genomics, etc. Or you can go towards biostatistics. So those specializations come under the electives. So there are core courses, and then there are electives that you have to cover, and then you have sufficient chance to pick among different subspecializations even within digital health.
Yeah, thank you, sir. Professor Pramod, you would like to add anything?
Yeah, so we looked at the leading universities and their curriculum, which offer health informatics or digital health type of programs at a master's level. And we have taken the best of what is offered at, let's say, Johns Hopkins University or Stanford University. So many of these leading universities have health informatics or digital health programs where the program in general, regardless of where it is offered, it is typically an interdisciplinary program where doctors, biomedical scientists, and computer scientists, data scientists have to come together and offer programs that will that will create a new breed of engineers who have that right orientation to serve the healthcare industry, hospitals, health insurance sector, pharma companies, and so on. So it's at that in that in mind. The other thing that has been kept in mind is that Government of India has recently launched a program called ABDM, which is Ayushman Bharat Digital Mission. Is that right, Amit? The name?
Yes.
ABDM. So
Right.
it's a digital. It's basically the Government of India's effort to digitize healthcare, health delivery, the diagnosis, and so on. Some of the coursework and so on has been designed keeping that in mind. So to create the next generation technocrats who will be able to participate in that whole process.
All right, thank you, sir. So, the next question is on the internship opportunities that KCDH has been offering to the students. Can you just elaborate on those as well?
Yeah, so of course, IIT Bombay has an internship, the placement office, which facilitates internships. But we collectively, the faculty of KCDH and the KCDH office itself, has developed several contacts in the industry, in hospitals, and so on. So in the relevant sector, so we try to create internship opportunities for our students, both the minor students as well as IDDP students. And several of our current batch, the students from current batch, have availed that opportunity. And these are typically maybe Smita or Rashmi can give examples of what are the kinds of companies that have been coming to offer internships. It may be, so the students are currently undergoing internship. This is the first batch of students who is having internship right now. It's too soon to see whether they will convert into a PPO, but there are good chances that some of these will convert into a PPO. So can you, Smita, can you give examples of the companies which are giving internship right now?
Yeah, currently we have on board ICSR Lombard, Bajaj Insurance, as well as actually IQVIA company is also offering, PwC is offering in fact, and even GE Healthcare we are in talk with. So kind of we are actually trying to reach out to actually healthcare sector, and so this way actually we are trying to get connected to them, yeah, even for internship as well as placement.
Right.
Yeah,
So they range from medical image analysis to healthcare, insurance companies, and also hospitals themselves. So these are, I mean, a broad range related to healthcare.
Yeah, even ThoughtWork is on board actually.
All right, thank you, ma'am and sir. So, any final remarks that both of you would like to give to the students who are interested to apply for IDDP, especially since the IDDP applications will be opening up very soon?
So uh, my final remark would be that this is a very exciting field, in the sense that it has a great impact on human well-being, of which we need a lot of improvement, especially in our country, in healthcare. And we don't have sufficient people who are trained in an interdisciplinary manner. Our education system evolved to have these silos of engineering and biology and medicine and so forth. So this is our chance to participate and innovate at the intersection of some of these fields and make direct impact in people's lives through digital health.
Yeah, I agree. I think this is an exciting field. There is definitely shortage of trained manpower in the field, and the companies are really struggling to find people who are oriented toward both these areas. There are people in one area, either the healthcare area or the informatics area, but health informatics is a new field, which is I feel an exciting opportunity. The IDDP program offers that immersive experience which you have not experienced. In my opinion, you may not have experienced that at IIT in the BTech program. So this is definitely something exciting.
Thanks a lot, sir. It was really insightful. Thank you for making it here to the session and conducting it.
Thank you. Bye-bye.
Thank you.
Thank you.
Thank you all.